In the early 1990s, the parish of St. Mary operated like any other Catholic church in Buenos Aires. The congregation attended daily Mass, entirely unaware of the biological anomalies about to unfold. The sequence began in May 1992. A Eucharistic minister found fragments of a consecrated host and placed them in water to dissolve, standard procedure for disposal. Days later, those fragments had turned a deep reddish color. The physical anomalies escalated over the next 4 years. In July 1994, unexplainable drops of blood appeared on the communion vessels. Then, in August 1996, a host that had fallen to the ground and been placed in water transformed entirely into a blood-like substance. The parish stored the initial 1992 samples in distilled water. Biologically speaking, this is a destructive environment that typically accelerates cellular degradation and dissolves organic tissue. Curious, yet when a local chemist analyzed the sample, she discovered active human white blood cells. These cells possessed a complete leukocyte formula, a biological defense mechanism that typically appears in the presence of a live infection. Genetic testing in 1992 lacked the precision to explain the cellular activity. To resolve the mystery, the local archbishop, Jorge Bergoglio, who would later become Pope Francis, authorized a formal forensic inquiry. The parish's claims were no longer matters of faith alone. They were now the primary evidence in a biological investigation. By October 1999, Australian lawyer Ron Tesoriero arrived to structure the investigation. He applied the strict rules of a court proceeding, treating the miracles as a legal cold case, requiring an airtight chain of custody. The team documented every step. They extracted samples, sterilized them, and sealed the containers on film to prevent claims of contamination. A forensics lab in San Francisco analyzed the tissue in 2000. They confirmed the presence of human DNA, but they could not extract a usable human genetic code. The medical community remained fractured. Early assessments, like that of Dr. Robert Lawrence, concluded the sample was human skin tissue. Standard genetics verified the sample was human, but the conflicting pathological reports left the case unresolved. Investigators realized they needed a world-class, blind medical ruling to eliminate bias. In 2004, the evidence arrived at the New York laboratory of Dr. Frederick Zugabe, a leading forensic pathologist and cardiologist. He examined the microscopic slides blindly, without any prior knowledge of their origins. Dr. Zugabe identified the sample as myocardium, specifically muscle tissue from the wall of the left ventricle. The heart muscle was severely inflamed and had lost its natural striations. Most critically, it was heavily infiltrated by white blood cells. In human biology, white blood cells only rush into muscle tissue to address trauma inside a living body. Dr. Zugabe mapped the specific cellular distress. He concluded the heart had suffered a severe lack of oxygen, consistent with a heavy blow to the chest over the heart. Analyzing the state of the cells, the pathologist provided an exact forensic timestamp. The traumatic injury occurred exactly 3 days before this microscopic snapshot was captured. He was then informed the tissue had sat in distilled water for years. Zugabe noted that it was scientifically impossible for the white blood cells to be so perfectly preserved in such conditions. They looked as if they had been kept in formalin. Presented with this blind pathology, previous skeptics re-examined the slides. Dr. Lawrence reversed his initial identification and agreed with Zugabe's assessment of traumatized heart tissue. Blind science had documented a medical paradox: a living, traumatized human heart, preserved for years in conditions that should have destroyed its cellular structure. The clinical reality of a beaten, living heart stands in sharp contrast to the quiet environment of a Catholic Mass, where a priest elevates a simple white wafer. Yet, this forensic diagnosis aligns with the core theology of the Church, which defines the Eucharist as a physical memorial of Jesus Christ's trauma and death. Catholic doctrine maintains that receiving communion is receiving Jesus at the exact moment of his resurrection. Looking at this dual timeline graphic, the medical findings converge with the historical text. Dr. Zugabe's blind forensic timestamp, showing a trauma that occurred exactly 3 days prior, perfectly mirrors the timeline of the resurrection, which takes place 3 days after the Passion. Ron Tesoriero concluded his investigation where he began, treating the matter as a legal cold case. He found that by applying strict forensic standards, the data of a 3-day-old trauma and the theological timeline of the resurrection had arrived at the same point.